Thank you very much, Michelle. Hello, everyone. My name is Prakash, and it's a pleasure today to talk about my PhD research on high-dose inhaled powder formulations of rifampicin, their preparation, in vitro characterization, and in vivo evaluation. Despite the availability of potent oral and injectable drugs, the treatment of tuberculosis faces challenges due to suboptimal concentrations of the drug in the systemic circulation and the lungs, where the bacteria mostly reside. Therefore, better treatment regimens for TB are always desired, and to become more efficient than the current regimen, the treatment strategies should include delivery of a high dose of the drug or achieve high drug concentration at the target sites within the body. One such method is the inhaled therapy that can achieve high drug concentration in both local and systemic sites after drug administration. Here we present an example of rifampicin, a first-line highly effective anti-TB drug. High-dose rifampicin is popular in oral drug delivery that refers to the higher than standard oral doses of rifampicin, that is more than 10 milligrams per kg per day. However, it is associated with increased risk of systemic toxicity of the drug, and inhaled rifampicin therefore can be an alternative to high-dose oral rifampicin for efficient treatment of tuberculosis. Two different approaches were used to prepare rifampicin particles for inhalation in this study. The first approach was spray drying, while the other was crystallization technique. 4 different powder formulations of rifampicin were obtained. Rifampicin A was obtained by spray drying, and rifampicin C1, C2, and C3 were obtained by crystallization. The crystalline nature of the powders was studied using X-ray diffraction technique, which confirmed that rifampicin A was amorphous, while rifampicin C1, C2, and C3. Upon in vitro aerosolization using NGI, it was observed that all powder formulations had fine particle fraction higher than 50%, suggesting good aerosolization potential of the powder formulations. Among the powder formulations obtained, the amorphous and the crystalline dihydrate powder formulations were selected for further in vivo study in a rat model. After intratracheal administration of the powder formulations, the effect on the lungs was evaluated by histopathology, and the effect on the liver was evaluated by serum alanine transaminase ALT assay. And the pharmacokinetic evaluation was done on day 0 and day 6 after repeated administration of powder formulations on once daily basis. The histopathological evaluation of lung sections obtained after 7 repeated administration of intratracheal rifampicin suggested that there was no toxic effect of the drug on the local tissues. The architecture was similar to that from non-treated group and the oral rifampicin group. The results from the ALT assay suggested that intratracheal administration of rifampicin reduced the drug burden on the liver compared to the oral administration, and the effect of repeated administration is higher compared to the single administration in case of the intratracheal group. The plasma concentration time profiles of rifampicin after single and repeated administration of amorphous rifampicin powder formulations by intratracheal administration showed significantly higher plasma concentration of rifampicin in the intratracheal group compared to the oral group. The results were similar in the case of the crystalline dihydrate formulation of rifampicin. However, the plasma concentrations were lower compared to those from the amorphous formulation. This table here summarizes the pharmacokinetic parameters of rifampicin A. Similar to that indicated by the plasma concentration time profiles, the intratracheal 50 mg per kg group resulted in significantly higher AUC and Cmax in shorter time compared to the oral administration of the same dose. And the AUC for rifampicin after AUC of rifampicin after administration of rifampicin C3 was lower than that of rifampicin A, suggesting lower bioavailability of the crystalline formulation. In conclusion, both amorphous and crystalline dihydrate formulation of rifampicin were safe to rat lungs after intratracheal administration of 7 repeated doses. Intratracheal administration of rifampicin reduced the burden on the liver, while achieving significantly higher bioavailability compared to the oral rifampicin at same dose. Among the amorphous and the crystalline formulations, the amorphous formulation showed higher bioavailability. And before, yes,
I do apologize.
I'd just like to acknowledge my supervisors and the funding bodies before I end my presentation. Thank you.
Thank you very much, Prakash. Very interesting presentation. And actually, I was going to ask a question, but David Ferguson, again, who's obviously had his Weetabix this morning, he's actually very much echoed the type of question that I was going to ask. And I think it was very much about how do you take it further. So what I'm actually hearing is that you've obviously demonstrated some very positive indicative data from the rat studies. And the next question is, well, how would you translate that into humans? Because, you know, in an infected individual, they're going to have compromised lung function. How would you actually think about delivering the drug to the right parts of the lung when it can actually have the effect that you're looking to achieve for this molecule?
Yes, so first of all, the question about translation is a very important one, because we had always thought about translating these formulations and studying these in actual humans. So we had also planned a phase 1 study in healthy human volunteers, which was not possible due to some regulatory concerns, but we are still progressing towards the preparation of the study. So that will probably go ahead in a very close time ahead. So, and to deliver the drug to the right part of the human lungs, it is obviously the formulation properties that's necessary to the aerosolization properties that is important to be delivered to the desired sites in the lung, which is usually the deeper regions of the lungs where the caseating lesions of tuberculosis lie.
Thank you, Prakash. A couple more questions that have come in, and if I could maybe ask a practical question of you, in that the World Health Authority doesn't currently recommend single therapy for tuberculosis. Do you think this work, with inhaled rifampicin would change that, or do you see it as an add-on therapy to existing therapies?
Yes, exactly. Yes, and again, it's a very practical question that we had come across when we were planning these sort of clinical studies. And yes, because of those regulatory concerns, we can't really replace the oral therapy with inhaled one. But what we can do is we can start with adjunct or add-on therapy of inhaled rifampicin. So we can test in a group of patients where they're given the regular regimen, and they're also given a supplement of an inhaled dose.
Okay, thank you.
Thank you, Prakash.
And then one final question. Do you want to ask this?
Yes, more from a CMC perspective. So you were showing that the spray-dried material was amorphous. Did you do any stability? And what can you share about the robustness of the particle to environmental effects on storage?
Yes, we also carried out a preliminary stability study where we checked the changes in the aerosolization parameters upon storage for some time. So it was not a very long-term storage stability study. So, but during our short-term studies, we found that the amorphous and crystalline formulations both were quite stable, and there wasn't much change in the aerosolization parameters. But we definitely need to look at it on a long-term basis. But for a short-term study like 3 months, it was fine. And our aim was just to check our formulation for a very quick clinical study. So we had found good stability of the formulations.
Thank you. Over to you, Alex.
Well, Prakash, thank you very much for an interesting talk in the